Hey friends, welcome to this channel. If you're new here, I am Indrani from Bomb Biology, and in today's video we'll discuss about the protein folding in within the endoplasmic reticulum, and this is the series 4 or part 4 of this endoplasmic reticulum protein sorting video. So what happened in case of secretory proteins and in case of transmembrane proteins, how they are being translocated inside the endoplasmic reticulum lumen or in the endoplasmic reticulum membrane, that we have already discussed. Now, how the protein folding happens. The molecular chaperone BIP, that is also called as that is actually HSP70 or heat shock protein 70. This binds to the polypeptide chain as they cross the endoplasmic reticulum membrane, okay, and they actually facilitate the protein folding and assembly within the endoplasmic reticulum, and finally the folded protein is being produced. If there is any unfolded protein or misfolded protein, there are some other techniques for them, and we'll discuss it in a short delay. So among within this endoplasmic reticulum, there are several things going on, like disulfide bond formation. Now, in case of disulfide bond formation, we know that this happens within cysteine amino acid. Now, these bonds do not form in cytosol, which is characterized by a reducing environment that maintains cysteine residue in their reduced form, that means in their SH form. However, in the endoplasmic reticulum, an oxidizing environment promotes the disulfide bond formation, and these disulfide bonds formed in the endoplasmic reticulum play an important role in the structure of the secreted and cell surface proteins. Disulfide bond formation is facilitated by an enzyme called protein disulfide isomerase. The enzyme name is protein disulfide isomerase. Okay. Two other things happen. One is called N-linked glycosylation, and another one is GPI anchoring. So first we'll discuss about N-linked glycosylation. Yes, this is N-linked glycosylation. This N-linked glycosylation is a co-translational process. That means it happens as the translation is going on. And proteins are also glycosylated on specific asparagine residue. This is the specific asparagine residue or ASN residue. Now, oligosaccharide units consisting of 14 sugar residues are added to acceptor asparagine residue of the growing polypeptide chain as they are translocated into the endoplasmic reticulum membrane. The oligosaccharide is synthesized on a lipid that is called the dolicol, which is also a carrier protein, and it is associated or anchored with the endoplasmic reticulum membrane. The oligosaccharide is then transferred as a unit to the acceptor asparagine residue. Okay, and this oligosaccharide contain 2N-acetyl glucosamine, 9 mannose residue, and 3 glucose residue. This oligosaccharide is transferred from this dolicol to the acceptor asparagine residue, and after that this 3 glucose residue will be removed. Up to this part, the N-linked glycosylation happened in the endoplasmic reticulum. When this particular protein with this oligosaccharide group enter into the Golgi apparatus, there will be some other modifications, but up to this removal of 3 glucose residue, these things happen inside the endoplasmic reticulum or in the lumen of the endoplasmic reticulum. Now, how the GPI is being linked to the protein. Now, glycosyl phosphatidyl inositol, that is called GPI. Hope you can see whatever I am writing. So this is GPI. This GPI is glycosyl phosphatidyl inositol. This GPI anchors contain two fatty acid chain, an oligosaccharide portion consisting of inositol and other sugar residue. This part. Okay. The GPI anchors are assembled in the endoplasmic reticulum and are added to the polypeptides anchored in the membrane by a carboxyl terminal membrane spanning region. This part, I think you can see, this is the carboxyl terminal and this part is the membrane spanning region. Finally, what happened is the membrane spanning region is cleaved. This part will be cleaved and there will be a new carboxyl terminal end will be formed, which will join with this oligosaccharide part, and this is called the GPI anchored protein. Okay, so this is the GPI anchored protein. The main difference between this GPI anchored protein and this N-linked glycosylation is GPI anchoring happen post translationally. That means the translation procedure is completed, whereas this N-linked glycosylation is a co-translation process. That means the polypeptide synthesis is still going on. Now we'll discuss about how the glycoprotein is being folded correctly. We have discussed over here that the folding is done by BIP, and if there is any unfolded protein or misfolded protein, there are some other techniques how to remove them. So that thing we'll discuss in here. So what happened is, first the protein is entering and this part is the N-linked glycosylation. So glycosylated protein enters and there will be, as you can see over here, that cleavage of signal peptide and attachment of N-linked oligosaccharide occur while the protein chain is exiting the translocon. That means this is happening co-translationally and the 3 glucose residues will be removed, and then they will enter into the calnexin residue. Within this calnexin there will be folding. This calnexin actually helps the protein to get folded. After folding by this calnexin, the glycoprotein is released and the remaining glucose residues will be removed. And if the glycoprotein is folded correctly, then it will exit the transitional endoplasmic reticulum through here. But if there is any problem in the folding, then there will be the hydrophobic part or the hydrophobic residues of that protein will be exposed, and then it will be recognized by UDP glycosyl transferase. So that will transfer one molecule of glucose to that glycoprotein so that it again enter into the calnexin. So the cycle will go on. If in the next cycle the protein is folded correctly, it will exit the transitional endoplasmic reticulum. But if this happens that again and again this protein is misfolded or unfolded, then all the mannose residues, there will be 9 mannose residues, I think you remember. These 9 mannose residues will be removed and the protein will be translocated back to the translocon and it will enter into the cytosol where it will be exposed to ubiquitin. So polyubiquitination will be done and via proteasome that will be degraded into small particles. And in proteasome, this is a energy dependent pathway, so there will be ATP will be converted into ADP. So by this the glycoprotein folding occur within the endoplasmic reticulum. Okay, there are some other signals, about them we'll discuss in our next video.